Now let's take a look at lipids. Lipids are a structurally diverse group of molecules. They're lumped together in one category based on their solubility in water. All lipids are insoluble in water. Other than that, their characteristic shapes can be quite different. They're all nonpolar hydrocarbons, which is what makes them insoluble in water. As nonpolar hydrocarbons, that makes them hydrophobic. Hydrophobic means water-fearing. So hydrophobic compounds do not dissolve in water. Hydrophobic lipids in the fur of some aquatic mammals help protect them from the elements, as we see here at this otter. Let's take a look at some of the functions of lipids. Many lipids are for long-term energy storage. The energy that can be stored in lipid is a little bit more efficient than the energy we see in carbohydrates, because in carbohydrates we have a lot of oxygen in the molecule, which takes up a lot of the weight of the molecule. A typical lipid has a much lower oxygen content, so you can have a lot more carbon-hydrogen bonds that have a lot of energy at a much lower molecular weight. They also provide insulation from the environment for plants and animals. And they can serve as building blocks for other molecules, such as hormones. They also are important components of cell membranes. In this image we see here, all of these spheres with the little white tails coming off of them, those are a form of lipid, as are the little orange structures that are in between some of those. That's another type of lipid. Cell membranes are composed of different types of lipids. Let's take a look at the different classes of lipids. We have triglycerides, which can be either fat or oil. And the distinction between fat and oil depends on the fatty acids that are present in those molecules and whether or not they exist as a liquid or a solid at room temperature. That's how this is defined. So one that is a liquid at room temperature is an oil. One that is solid at room temperature is a fat. We also have waxes, phospholipids, and steroids. And we'll take a look at each of these. Let's first take a look at triglycerides, the fats and the oils. We have two main components to triglycerides. The first is a glycerol backbone. Glycerol is a 3-carbon compound we see pictured here. Then we will attach fatty acids onto where we see these OH groups here. So if we attach one fatty acid, which we see down here, we bring this up and attach it there. We have a monoglyceride. If we attach 2 fatty acids, so we would attach another one in this position, we would have a diglyceride. If we attach 3 fatty acids, we have a triglyceride, which is really more technically referred to as triacylglycerol. Each fatty acid that we add to the glycerol is a dehydration synthesis like we have seen before. So we take an OH group off of this fatty acid, an H group off of the glycerol, remove that water, and then create the bond between this carbon and that oxygen. So again, if we join 3 fatty acids onto that glycerol backbone, we have triacylglycerol. These linkages that we just created through that dehydration synthesis are called ester linkages. And again, it's between this carbon of the fatty acid and the oxygen that is coming off of this glycerol. So if we were to create a triglyceride, we would remove 3 water molecules and create 3 ester linkages. Let's look at the difference between saturated and unsaturated fatty acids. A saturated fatty acid means that it contains no carbon-carbon double bonds in the backbone. An example is stearic acid. You can see the backbone here, no double bonds in there. These tend to pack together very tightly because of all of these single bonds. These tend to be solid at room temperature and are likely to be associated with cardiovascular disease because of that solidity and should be limited in your diet. So again, we have all single carbon-carbon bonds. So the reason we call it saturated is because it is saturated with hydrogens. When we go to create a double bond, if I create a double bond right here, I have to remove some hydrogens to do that. And so if I remove some hydrogens, then it's no longer saturated with hydrogen, so it would be an unsaturated fatty acid. So let's take a look at that. So an unsaturated fatty acid will have at least one carbon-carbon double bond in that backbone. This one shows we have one in there. And notice that we have removed some hydrogens. If we have a monounsaturated fat, there's one double bond. If we have a polyunsaturated fat, there's more than one double bond in that chain. Notice we don't count the terminal carbon here in that double bond with oxygen. It's a double-double bond between carbons in the chain. Most of these will be liquid at room temperature, and they're referred to as oils, whereas those that have a lot of saturated fatty acid tend to be solid at room temperature and are referred to as fats. So here we have a double carbon-carbon bond in that backbone. It means that it's unsaturated because it does not have as many hydrogens as it could have. Let's take a look at the issue of trans fats. Why are trans fats bad for us? So at the top we have saturated fatty acid, all single bonds. The bottom two pictures we have unsaturated fatty acids that contain some double bonds. Remember, each of those double bonds can be in one of two configurations, what we call a cis configuration. Remember what cis means. It means on the same side. So here the hydrogens are on the same side of the carbon chain. We see they're cis, they're on the same side. Trans configuration, they're going to be on the opposite sides of the chain. So down at the bottom, we see the transformation with hydrogens on the opposite sides of that double bond. When we have the cis form, it forms a kink in the chain. And that kink in the chain means that you get more distance between chains. They don't pack as tightly, and it is more liquid. Down at the bottom, the trans fatty acids do not have a kink in the chain, so they form these straight chains, and because of that, they tend to be more packed more tightly and are more solid at room temperature. In processing of foods, many times they will take a fatty acid that is in the cis formation, and it will be converted to that trans formation, usually through heating and other processes. Now, the problem with that then is it makes it more solid, and it's not as healthy because any of these fats that are more solid at room temperature can contribute to heart disease. So why would they do that? Why would they take and process food where they're converting some of the unsaturated fatty acids from the cis to the trans formation? Sometimes it's done in order to get the proper consistency. You might want something that is a little more solid than liquid, say a candy bar, as opposed to having a liquid pool of chocolate. You might want it to have more of a rigid shape and texture. And so many times these trans fats are formed in that processing, and sometimes they are byproducts of heating. Let's take a look at what we mean by essential fatty acids. Something that is essential is required by the organism, but the organism cannot synthesize it on its own. Therefore, it must be part of the diet. So essential fatty acids are those that we would have to consume. We also heard a little bit about omega fats. Let's take a look at what omega means. All omegas are polyunsaturated, so we have lots of double bonds along those chains. When we look at this fatty acid, we will label the last carbon in the chain as the omega carbon, or the omega end of the chain. We have our first carbon here next to that double bonded oxygen, all the way down the end of the chain. This is our omega end. When you look at a fatty acid that has a name and number, omega-3, omega-6, omega-9, the omega is telling you where the first double bond is if you count from the omega end. And this position will determine the shape and function of that molecule. So omega-3s will behave differently from omega-6s, which behave a little differently than omega-9s. The fatty acid we see here is omega-3 because the first double bond on carbon is occurring at the third carbon away from the omega end. Omega-3 fatty acids tend to be found in salmon, trout, and tuna, and also in flaxseed and walnuts. This is an omega-6 fatty acid. It will have its first double bond between carbons on the sixth carbon from the omega end. So this is our first carbon. 1, 2, 3, 4, 5, 6. This is our sixth position of carbon from the omega end, so this is an omega-6. If we didn't have this double bond here, and this was our first double bond, then we would have an omega-9. Omega-6s are found in vegetable oils. These fats are heart healthy. These are fats that are good for us. They reduce the risk of heart attack, they reduce triglycerides in our bloodstream, and they tend to lower blood pressure. Both of them are beneficial. However, it's likely that our modern diet contains the wrong ratio of omega-3 to omega-6. Historically, throughout our evolution, we probably had a lot higher omega-3 and a lot lower omega-6 in our diet. Now we have much higher omega-6. Some studies suggest that maybe we need to be careful with this ratio of the omega-3 to omega-6 and make sure that our omega-3 ratio is higher than the omega-6. Some studies suggest that omega-6 can contribute to inflammation, and so you may read in some places where they're telling you to avoid omega-6. Other studies show that omega-6 is probably not a big contributor to inflammation, so the jury is still out on whether omega-6 really significantly contributes to inflammation. But what does seem to be clear is that we should at least have a higher omega-3 than an omega-6 intake of these fatty acids. Let's take a look at waxes. We see here the formation of a wax. We take an alcohol and add it to a fatty acid to create a wax. This process is called esterification, when we take these long alcohols and add it to the fatty acid. They are also hydrophobic, lots of hydrogen-carbon bonds, which are going to be hydrophobic. It prevents water from sticking to the surface where you have this waxy coating. We see this on the feathers of some aquatic birds and also on the surface of some leaves. Let's take a look at phospholipids. Here we see 3 different ways that we can look at a phospholipid. Here we see the structural formula where we're seeing the carbon chains and the other atoms that are in the other groups here. This is a space-filling model trying to show us the position of those atoms. Here we have more of a symbol of a phospholipid, and it's done that way a lot of times when we're drawing pictures of like cell membranes, because there's so many of them, you wouldn't want to show all the detail of the rest of the structure. Phospholipids consist of a glycerol backbone. That's what we see right here, is that 3-carbon compound like we saw in our triglycerides. We're going to attach 2 fatty acid chains. Basically, we create a diglyceride here. And then on the 3rd position of the carbon chain, we add a phosphate-containing group. So here we have phosphate. And then the phosphate can be modified. And when you see R is usually representing a variable portion to a molecule. That basically is telling us we can add some other stuff there. And it can be a variety of different things that we add. And it could be a long chain or whatever. So it can be a lot of different things. And so we add these R groups to modify this basic phospholipid. There's 2 common chemical groups that we see attaching to this phosphate group. One is choline, and it creates phosphatidylcholine. So we can see the choline component right here. Here's our phosphate group, and then this piece here that is attached to the phosphate group is choline. And then this whole molecule is called phosphatidylcholine. We can also attach serine to it. So here is our phosphate group. Over here is this serine group. And this creates phosphatidylserines. Let's take a look at the function of phospholipids. Phospholipids make up the major component to our plasma membranes. So here's our phospholipid molecule. First, let's talk a little bit about the molecule itself. It's an amphipathic molecule. What that means is part of it is hydrophobic and hates water, and part of it is hydrophilic and loves water. If we look over here at the molecule, we can see these long fatty acid chains. We know that those are going to be nonpolar. That means they're going to be hydrophobic. They're going to want to be away from water. Here we have the phosphate group, and this phosphate group is a charged group. That means that it is attracted to water. Remember, anything that is either polar or charged likes water, because water has those partial charges on it and it's attracted to that. So this negative here would be attracted to the water molecule. So this part of the molecule likes water. This part doesn't like water. So we have this polar head that is hydrophilic. It will tend to face a watery solution. We also have these hydrophobic tails that will tend to go away from water. So in a membrane, if you think about your cells, they're basically little fluid-filled sacs that are floating in fluid or surrounded by fluid. Imagine that there's water all the way around this. These polar parts of the molecule, these hydrophilic parts, are facing the water, and then these tails, which are hydrophobic, point inward towards one another. So they sequester themselves away from the water. And they form this middle bilayer that is hydrophobic. So this whole area in here is hydrophobic, but the two outer sides here near these polar heads are hydrophilic. These phospholipids then contribute to the dynamic nature of the plasma membrane. They can move their position, and depending on the exact nature of those fatty acid chains, can make this membrane more or less solid. The degree of saturation of those fatty acids can vary from one type of organism to another, creating differences in how solid or fluid that membrane is. Now that can be very important, we'll see later, in how membranes function. Your membrane needs to have a certain level of fluidity and a certain level of solidity. It needs to be just perfect, right in the middle. But what it takes to create that depends on, for one, the temperature the organism lives at. So if I have an organism that lives at very cold temperatures, I might need a different fatty acid composition to keep that membrane just right than if I have a tropical organism living in a very warm environment. So different animals and plants could have different membrane structures and different fatty acid composition in their membranes in order to achieve that perfect amount of fluidity in their membranes. Let's take a look at steroids. Steroids are different from the other lipids. They have a closed ring structure. What we see here are 4 interlocking carbon rings. So all of your steroids will start with that basic structure of those 4 interlocking rings, and then we can add other things onto it. So we see something different added right here on cholesterol than what we saw over here on cortisol. Many of them will have these short tails on them. And one of their characteristics is that, as you might imagine, since they are these large hydrocarbons, they tend to be hydrophobic, meaning they are also insoluble in water. The most common steroid we find is cholesterol, which is what you see on the top there. Cholesterol is synthesized in the liver. It is another component of cell membranes. So not only do we have phospholipids in our membranes, but we also have cholesterol in our membranes that helps to stabilize them a little bit. It's also a precursor to other hormones like testosterone and estradiol. It's a precursor to vitamin D. It is also a precursor to bile salts. While we often think cholesterol is bad for us, obviously there are some very vital functions for cholesterol. It's just that certain types of cholesterol in our bloodstream can lead to heart disease. So that gives us an overview of lipids.